Hello everyone, in this video we're going to talk about something called the test cross. It is also a monohypercross, but it adds a little bit of a twist to it. So if you can remember, when we talk about monohypercross, it is only one trait. So here we're going to look at an example with a fly, fruit flies. Okay, so this is, there's no practice at home, but just think about this. If we have a fruit flies, there's one trait that we're looking at. We're looking at the eye color of the fruit flies. There are two possible variations of the eye color for the fruit fly. Remember, whenever we have a variation of a gene, we call that an allele. So the dominant allele is red. So red eyes are dominant, or the red eye trait is dominant, and the red eye allele, which is big R, is dominant over white eyes, which is little r. And what we're doing is we're crossing a white-eyed fly with a red-eye fly. And we could also cross two heterozygous flies. We could do whatever we want, okay? But here's the problem. Knowing that red eyes are dominant over white eyes, here's the problem. Can you determine the genotype of a white-eyed fruit fly just by looking at it? So if you look at this fruit fly right here, we said, again, the white eye is recessive, okay? So think to yourself. Are you thinking? Can you determine the genotype just by looking at it? Yes, you can, because if we know that white eye is recessive to red eye, the only way that a fruit fly can have white eye is when you have two recessive alleles. Remember the wording. Some people are still confused about genes and alleles. So if we were to have two recessive alleles, this is going to be expressed as little r and little r. Okay? So yes, little r, little r. Okay, write that down on your notes. And make sure you understand what's going on. And now the second question is, can you determine the genotype of a red-eye fly just by looking at it, right? So red eye is dominant over white eye. And if you think about it, there are only three possible genotypes of an individual. There's big R, little r, and little r, little r. Which one of those three, or which two of those three, have the trait of red eye? So it could be big R, or big R, little r, right? Both of those genotypes would give you the phenotype red eye, right? So how would you be able to determine the genotype of a red eye fly? Because whenever you look at a person or a fly or whatever organism that you're looking at, you can only see what you see. You can only see the phenotype. But how would you know the actual genotype? Right? If you have a recessive trait, you can automatically know what the genotype is, but you cannot know that for a dominant trait. So what do? You perform something called a test cross. Here's the magic of a test cross. So you cross two organisms during a test cross, just like any kind of crosses. You need two organisms, but there's something special about this two organisms. It will always involve an unknown red eye, so an unknown dominant individual with a homozygous recessive individual. So you have this question in your head. We have this red eye fly. Is it big R, big R? Is it big R, little r? And you're going to purposefully cross this unknown genotype for a dominant phenotype, right? With a homozygous recessive the trait individual, or you have the recessive trait, okay? So the white eye fly, little r, okay? So if the red eye fly was big R, what would the offspring of the test cross look like? So on your paper, there's this Punnett square. You want to do the Punnett square of big R, or big R, little r. You don't know which one it is, so we're going to try big R first, but we are always crossing it with little r, and what happens? So if you were to cross it, you're going to see a Punnett square with all the offsprings with the genotype of big R, little r. So do the Punnett square on your paper, okay? And what you will find out is that all the offspring of this unknown red eye with the white eye fly would have red eyes if the red eye fly was actually big R, okay? But if you were to try the Punnett square, so do the Punnett square on your paper, pause the video, do the Punnett square. If the red eye fly was big R, little r, you purposefully cross that with a white eye fly, so that has to be little r. When you do the Punnett square, you're going to see half of the offspring would have big R, little r, and half of the offspring would have little r, little r. So actually confirm that with your Punnett square and see if that's what happens. So if half of the offsprings are big R, little r, those big R, little r offsprings are going to have red eyes, and then those little r offsprings are going to have white eyes. So what's the point of this? Okay, you have an unknown individual. You know the characteristic of this fly is red eye. And you cross it with this white eye color fly. You cross those two, and now your job as a scientist is to look at the babies. When you look at all the offspring fly, all the baby flies, you look at each one of their eyes, and then you see if okay, so here's the result. If what you see, all the offsprings, if all of them have red eyes, that means that unknown individual parent with the red eye has to have homozygous dominant genotype. Big R, big R. All right? Let me say this one more time. Think in your head. Make sure this makes sense to you. What was I saying? Okay, so you're crossing an unknown red eye with a known white eye, because when you have white eye, you always know it's little r, little r. When you cross those two flies, you look at all the babies, and when you look at all the babies, if all of them have red eyes, that means the unknown parent with red eye has to have the genotype big R, or you can also call that homozygous dominant. However, if when you look at all the offsprings, all the baby fly, you look at it under a microscope, you count, look at the eyes. If the result is you have some of the offsprings that have red eyes, some of the offspring having white eyes, that means the red eye unknown parent has to have the genotype big R, little r. Okay, so that's the deal. That's the whole point of text cross. You're trying to figure out, well, we have this dominant phenotype. What is the genotype? You look at the offspring. Okay, so we talk about dihybrid cross already. Just one last thing. So if you think about this white-eyed offspring, anytime you have an offspring that has any genotype at all, right? This offspring could be big R, little r, in other scenarios, right? Whenever you have two letters, remember this, you have to remember this. A lot of people got this wrong during the test. When you have two letters, those two letters represent two different alleles. Big R, little r, you have a big R allele and you have a little r allele. What's the point of this? Those two alleles come from the two parents. Big R comes from mom or dad, little r comes from mom or dad. So let's say you were to have flies with white eyes, little r, that means one of those little r comes from mom, one of those little r's come from dad. So the offspring always gets one allele of each gene from mother and one allele of each gene from dad. That's the case of all these Punnett squares that you've learned, okay? So you have to understand the logic behind it so that when you come to AP Bio, you'll be so good at Punnett squares, because a lot of people are still confused by the time they get to AP Bio, okay? One last thing. Here's some numbers for you to remember. Whenever you have, if you remember some of the crosses that you did, okay? Whenever you have two heterozygous parents, you would always have 3-to-1 phenotype, okay? So you can do a heterozygous cross right now. Big R, little r, you're going to have big R, and then two big R, little r, and little r as the offspring. That will give you 3-to-1 phenotype. 3 for the dominant, 1 to the recessive. Monohybrid, another one is whenever you're doing a test cross with a heterozygous and a homozygous recessive, the one that you just did just now, big R, little r crossing with little r, you're going to have 1-to-1 phenotype, right? Half of the offsprings are going to have the dominant trait, half the offsprings are going to have the recessive trait. Last one, whenever you do a dihybrid cross where you have both parents that are heterozygous, remember the one we did in class one of those days, Thursday, if you have big R, big Y, little y, crosses with big R, big Y, little y, again, you would have 9-to-3 to 3-to-1 ratio for the phenotype. So these are just some numbers for you to remember. It's good for you. These are numbers that are really important when you come to AP Bio a few years later. This is going to be really good for you. Okay, so now here's a feature of Marshy. Here's Marshy the rabbit. He was really proud of all the Punnett square that you're learning.